Personally, what motivates me is developing new models which will better mimic cancer. So using cells that are directly derived from the patient. And what I hope from this consortium is learn new knowledge on persister cells and apply this knowledge on the clinic to improve patient treatment.
What drives me is the opportunity to bring our lab-based knowledge to the clinics in order to treat the patient.
My name is Clara Fauveau, and I'm currently a third-year PhD student. And inside Transgene, I'm working on developing in vitro models based on patient-derived cells to be able to better understand persistence and also trying to define if transgene product could target these cells.
I'm Shreyansh Jain. Currently, I'm team leader of open innovation and microphysiological systems in Transgene. I'm also a supervisor of Clara, where I'm trying to guide her on establishment of persistent models inside the lab.
So inside the consortium, Transgene has been working on developing new models to be able to study persistence, giving these models or these samples to the consortium partner, which is able to analyze them at the single cell level. So we've been generating some new data on persistence, and I've also been participating to different work packages to try to discuss the persistent signature and try to put in place methods to analyze the data in a common way across the consortium. What makes Transgene contribution unique inside the consortium is the use of 3D in vitro models using patient-derived cells. And the idea behind this use of patient cells is to have more complex models which better mimic and reproduce what happens in clinic. And the other advantage of Transgene is that it has a portfolio already of products which are designed to target cancer cells, and we're trying to identify which products are the best to tackle persistence.
We are the only consortium partner who is trying to work across 6 different NSCLC, which is non-small cell lung carcinomas, patient samples. What Transgene does is use viruses to treat the cancer. Viruses is being used in 2 formats, one as a therapeutic vaccine, and the second format is using them as an oncolytic agent. We already start to see that these viruses could eventually be an effective way to tackle the challenge of persistence. These oncolytic viruses are, let's say, a family of viruses who have more liking towards cancer cells. In a simple words, they can breed more specifically inside the cancer cells. Now, doing so, because of an intensive virus breeding inside the cancer cells die. Transgene is using these engineered viruses to specifically target these cancer cells. And in this context of our consortium, we are trying to see if these viruses can also be very effective in treating the persistent cancer cells. This consortium is very unique in tackling the challenge before it worsens in the patient.
Since the beginning of the project, we've generated data on persistence with cell lines. We've analyzed the signature persistence of lung cancer treated with chemotherapy. And so we've also identified that transgene's oncolytic virus are able to tackle persistence in this model. Now we're moving on to a more complex but also more relevant model using patient-derived cells. So we've created a biobank of lung cancer cells. We've established protocols to culture them and also to induce persistence. And so the samples are ready. They are now getting analyzed at the genetic and transcriptome level. And so we're eager to have the results and analyze them.
What we rely on for this project is 2 major technological platforms. One of the technologies is built inside internally in Transgene, where we create this 3D architecture of tumors, which is really mimicking a tumor inside a human. They are very specific in terms of how cancer cells are organized inside or on the external side of the tumor.
So we're trying to recreate microtumours. They can be made from cells that we've obtained from samples of patient biopsies.
Now, another part of the technology is rather coming from the consortium side, where we are using single cell transcriptomic based analysis for all our models across the consortium.
The consortium provides single-cell methods to try to analyze the persistent signature inside each of the cells of our model to try to see if persistence has common mechanisms or different mechanisms inside our models and across models.
It gives us a deeper insight into how heterogeneous a tumor could be and how, after the chemotherapeutic treatment, the transcriptomic profile of the cancer cells evolve. This helps us know what exactly to target and how our targeted approach of putting a therapeutic candidate might have altered the state of the cancer cells. Either it could be in a good way or a bad way. Something which could be very interesting from Transgene's perspective and from the perspective of consortium is to see if different patients could have a different way of persistent cancer cells being established after the chemotherapeutic treatment. Using patient samples comes with a lot of responsibility. And here in Transgene, we ensure that we have all the right permissions in place when we use these samples. Something which is very important for us because we work with patient sample is to ensure we leverage from each and every part of the patient sample which we receive. Every microtumor we grow inside the lab represents a person in the clinics. And that's always going through our mind when we handle these samples and we conduct the experiments. In fact, when Clara works with these samples inside the lab, we ensure that all the part of the sample are being used for one or other analysis. Nothing goes to waste.
I think it's been great to collaborate with partners across Europe. We have the annual meetings, which took place in Cambridge, here in Strasbourg, in Utrecht as well. And it's been a great way to meet in person the different partners. Know that the consortium is going more toward analyzing the data more than the setting up. It's been a great experience to be able to analyze, compare, and discuss results. And it's been a great way to have help and advices on how to handle and analyze our results, and putting it all together will, I think, introduce new ideas in the field of persistence. What I'm the most excited about for the remainder of the project is to start the analysis of patient cell data. So I've prepared the samples, they've been shipped to different partners for analysis. And so in a few weeks, I will receive the data and start the analysis. And so it will be the first time inside the consortium that 6 different patients from the same indication have been used as model to study persistence. And so I'm really eager to dive into the data and present my result to the consortium and have their feedback.
One of the final part of the project, Transgene has created microtumors from 6 different patients. Now, all these tumors which we have established inside the lab have been treated by Clara with chemotherapeutic regimes. These samples are being now analyzed for single cell transcriptomics, and we are pretty excited to receive these data sets back. And we are eager to see if we manage to pull out or dig out some persistent specific signatures across these patients. Now, very important aspect for this project will be to match the single cell transcriptomic data of each patient with the oncolytic virus treatment which we have done for these patients. We are pretty much looking forward to see if we can find some relevant biomarkers of the patient where our oncolytic virus treatment would have worked more efficiently.